Hello, everybody. It would never be a proper stream with me if I didn't forget to turn on the microphone. How is everybody going, by the way? Yeah. You should hopefully be hearing me now, so let me know if... Hello, can everyone hear me now? You should be able to. Yay, sound. All right, awesome. Again, it's traditional. I always forget to check the sound before I start. Okay. Yeah, I love it when OBS changes my settings without letting me know about it. It's just I'm so shit at everything when it comes to streaming. Okay. All right. So anyway, how is everyone? I hope everyone's all good. I'm back at home because I actually have internet at the moment because it's changed over. All right. So first of all, I want to say hello to Surus, who I think's just popped off. He popped in, said hello, and ran away. Ravenhorn, Sam Chaos. So basically 411. And I think that's everybody that's in. Oh, Dotty Little, hello. And Dot Nerd. And Danish Debater. So asks a good question. What is today's topic? Today I got a fairly much easier to understand research paper than from last week, which is talking about Salmonella species that were found in viviparous species of snake. They chose the two species of pit vipers. Sam Case says, I'm doing pretty well, bar body pains, but that's what painkillers are for. Love a late evening stream or, late evening over on Turf Island. Turfs. Yes, I do not like turfs, by the way, in case you're wondering. I'm not a big fan of them. And Ravenhorn says, it's a day is all. Nerd says, oh my God, I have 99 pings on here now. I'm not entirely sure what you mean by that, but if that's a good thing, yay. If it's not a good thing, not yay. Okay, so this week's paper is pretty interesting. It is called The Analysis of the Transmission of Salmonella Species Through Generations of Pet Snakes. So this one's a little bit easier to understand than last week's. So I'll be able to read it through with you and have a bit of a chat about it. I found it absolutely fascinating, so that's why I chose it. I'm actually going to be concentrating on E. coli at uni, but when I read this, it was like, oh, this is cool. I got to tell everyone about it. So, all right. I'm going to see if I can try and not break OBS. Actually, no, I'm not going to try and break OBS. I'm just going to do this. So I can see what's going on. Okay. So, the summary, besides the classical animals known as reservoirs for Salmonella species, Salmonella is, of course, the species of bacteria that causes you to get food poisoning, and it's not an pleasant infection, as Sam Chaos said. Salmonella is unpleasant. My grandmother, I think, had salmonella poisoning once, and she said she couldn't eat chicken for a good couple of years later. She was so sick. She was really, really sick. Danish Debater is OBS is broken. OBS is broken in the way it likes to break other people. I think this is intentional. I think the people who made OBS like this wants to hurt people. I think they're actual sadists, as well as the fact that we are masochists for using it. Anyway, so hang on, where are we? Reptiles have emerged as a significant source of human salmonella infections during the last years. Reptile-associated salmonella frequently cause severe clinical causes severe clinical causes including fatalities due to septicemia and meningitis. Meningitis is the inflammation of the meninges, which are like a thing in your central nervous system. It makes you very, very, very sick, as most people would understand. Therefore, it is of major priority to develop measures which may help preventing cases of reptile-associated salmonella. However, as a first step in the epidemiology of salmonella in reptiles must be understood. Therefore, in the study, a population analysis of the salmonella of 2 female snakes was performed, and the pattern of inheritance of salmonella to their offspring was investigated. Sam Chaos, OBS developers are clearly sentient salmonella trying to invent new ways to bite us besides snakes and chickens. Okay, so basically what they're trying to figure out is trying to see how salmonella species are being introduced into snake species, which within, as we know, within epidemiology, we know that it is pretty important to know where the source of something is to stop people from getting sick. Okay, so it's really interesting that the colonization, it is demonstrated that adult snakes usually harbour a population of concurrent Salmonella cerevars. Colonization of their offspring during pregnancy and birth is a significant way of transmission, causing 65% of the newborn to be positive for Salmonella. The effectiveness of the transmission does not seem to be due to only the frequency of certain cerevar, because the most prevailing strain of one female snake was not detectable in any of her offspring. Which I thought was pretty interesting. And if I go... Okay, so what there was, the introduction is basically saying that because snakes are becoming more and more .NET says, I already feel queasy today. I don't know if this will help ROFL. Hopefully, well, we're not going to be showing any disgusting pictures or anything like that. That's for later. That's for the shorts that I put out. So basically they're saying snakes as pets become really, really popular. And so what they're trying to figure out is if there's like they're noting that during recent years the During recent years, the CDC has repeatedly issued evidence for an increasing number of reptile-associated salmonellosis in the USA. Different reptiles such as turtles, iguanas, chameleons, and snakes have all been identified as possible sources for human infections. And the main serotypes isolated from patients with reptile-associated salmonellosis include Salmonella enterica species Diarrhoezonae 3B cerevars, the Salmonella enterica species Hutinae cerevars, Camellia and Marina, and S. enterica species Enterusiae cerevars Java, Stanley, and Puna. So basically these particular species are the ones that get you really, really sick. The most common one, from what I remember when I looked this up, the most common one that causes people to get really sick is the Salmonella enterica species Diarrhoezonae cerevar. Right, this one. What is a cerevar? I did look that up, but I didn't quite... what is a cerevar? I think it was... according to the Britannica, go away, pop-ups. Give me a moment, it's coming. Cerevar is a different strain, basically, from what I'm seeing. So a cerevar example. what is a serovar? Or a serotype or a serovar is a distinct variation within a species of bacteria or virus among immune cells of different individuals. A group of serovars with common antigens is called a serogroup, or sometimes serocomplex. Serotyping often plays an essential role in determining species and subspecies. Yeah. So that is what a serovar is, or a serotype. So the main serotypes isolated from patients with reptile-associated salmonellosis include... hang on, I've already read that. In contrast to other salmonella, these serotypes often cause severe clinical courses, including fatalities due to septicemia and meningitis, especially among children under the age of 10 years and immunocompromised people who seem to be especially prone to infections with reptile-associated salmonella species. And it has been demonstrated recently that reptile populations are colonized with salmonella in very high percentages. However, it's still unclear whether the snakes become colonized by contaminated food or whether transmission occurs during pregnancy and birth. So, the thing is, they decided to ask that question: where do they get their salmonella species from? And I'm just going to go down. I found... Go back. And so that is the table. See if you can... Oh, no, I can't make that any wider. Sorry, I'm just checking the chat very shortly. Oh, God damn it. Okay. So the chat, just going back onto the chat. Hi, hello, Todd. Wow, Todd, hope you stay safe. Hi, Mary. Sorry, I was having trouble. I was having to hide a storm shelter while funnel cloud rolled through. Just lovely spring weather here. Scanning to catch up. Soundcast, I've got to have to be ready for big words on your stuff, not just be a gremlin in the chat. Hopefully my avatar isn't carrying any of those species of salmon. Hopefully not. Stuck in Florida, hello. Thank you. Don't forget to hit the like button, everyone. That is a good point. Yeah, don't forget, like, share, subscribe, blah, blah, blah. You know the drill. Okay. So, as you can see with this, the females of the species, as I said, these are viviparous snakes. These snakes, viviparous means that they are born live. Humans, for example, are viviparous. Animals that are born from a shell, like from a shelled thing, like an egg, that's the word, a shelled thing. What the hell? If they hatch, they are oviparous. So, for example, my little gorgeous girls, they are oviparous. So the summary of the salmonella types found in the population analysis of fecal samples from 2 rhinoceros horned vipers and comparison with the serotypes found among their offspring. So the females, they had this particular serum, and they found it in their newborn. So number of isolates of this particular serum of female number 1, there was 2 species, 2 particular individuals. The first one did not pass this particular serotype onto their child, their newborn, although she passed it on these particular serotypes onto a newborn, and the newborn had a couple of other extra ones in here. And female number 2, she was able to pass on some other species of salmonella onto her newborn. So it says here, although this, in the present investigation, this number 3B38KZ Serotype was detected in the sample of female number 1. In a previous study performed 2 years ago, the serotype was found to colonize this particular snake. So they were able to, they had this particular serotype was not found in number 1, female number 1. They actually found it previously, so they probably didn't pick up any of that particular salmonella. And B, one of the newborns was simultaneously colonized by these two salmonella serotypes. Serotypings were performed by direct agglutination of microslides at the former National Reference for Enteric Pathogens. Generation of the antisera was performed by immunization of New Zealand rabbits following the respective WHO guidelines. Back here. Alright. So, go across. So what they did is they got a newborn snake. They just went, that's a healthy one. That's a good newborn snake. It's healthy. And they considered healthy in regards to its behavior. So reaction to environmental influences, drinking behavior, that sort of stuff. And uncomplicated sloughing or shedding. The first sloughing usually... Is it me or is slough a really gross word? It's just slough. The first sloughing usually occurs within the first few days after birth, and healthy snakes should shed their skin in one piece. That's basically the gold standard. Oh, also, talking about ugly words, most non-native English speakers find the word diarrhea to be a really beautiful word. And if you think about it, it's actually quite a beautiful word, but it's not a beautiful thing. So, anyway, fun fact for you. 20 newborn snakes met the outline criteria and were included in the study. Of these, 15, 6 male, 9 female, were born from female 1, and 5, 3 male, 2 female, were born from female 2. Although 13, 65% of them were found to be colonized with salmonella species, 5 of them carried an identical serotype as their mother. 14 different serotypes were isolated from the baby snakes, and all isolates belong to different serovars of S. enterica species Diarzinae, serotype 3b, with the exception of one isolate which belonged to S. enterica species Enterica serovar Corvallis. This isolate was identified in a newborn of female number 1. Summary of the isolated serovars is shown in Table 1, which is that table I showed you earlier. Only 2 babies of female number 1 carried salmonella of identical serovars as their mother. 2 additional babies carried serovar 3B38KZ, which was the predominant one in female 1 about 2 years ago when another study had been performed. Oh, thanks, Mike. I really appreciate that. Thank you for subscribing. Okay, so basically what they're saying is they're finding that there are the babies of female of their mother, the babies picked up salmonella from their mother at some point during the birthing process, or maybe even just the development. Stuck in Florida says, handling thousands of reptiles in my life, I have never knowingly had salmonella, although it's supposedly one of the most zoonotic diseases. It's pretty easy to catch it. It is a fairly nasty disease as well. But that's why you always wash your hands after handling your pets and stuff like that. I haven't got any salmonella from my girls yet, but I'm assuming that once I start getting the feces figured out and doing the plating and the DNA sampling and all that sort of stuff, there's probably going to be salmonella in there, I'd say. Apparently, you're most likely to get salmonella after they shed, because when snakes shed, they release an oil. to separate it, and that oil, of course, being thick, full of lots of nutrients and very wet, bacteria in that oil just goes absolutely crazy. Hello, Cyrus again. How are you going? I'm just going to see if you're coming up. Oh, yes, you are coming up. Excellent. Okay, so just going. Right. So they were talking here about live birth snakes, and they also looked into 12 non-fertilized eggs of a western gaboon viper, which were laid during a 3-day period, were examined for the presence of salmonella. These people like to dance with death, don't they? Cultivating bacteria from one of the most deadly animals on the planet, and then, oh yeah, let's just talk about salmonella. Like, let's just... Wow, these people, I would not be able to do that myself. Holy moly. I was reading that and just going, geez, these guys are brave. I wouldn't do that. So basically 411 asks, does the bacteria eventually go away, or do they stay in the snakes for all of their lives? So if you can guess, so can you, I guess, treat it, if that makes any sense? I would say they'll probably go their entire lives. I wouldn't bother, unless it's a really nasty salmonella, or if it makes people sick. I probably wouldn't treat for salmonella, mainly because if it's not making you or the animal sick, why would you treat it? If you don't need to give antibiotics. Excuse me. If you don't need to give any antibiotics or treat a disease, like it causes more harm than good. Yeah, so I would be surprised if they would do that. Okay. Additionally, 12 non-fertilized eggs of a western gaboon viper, which were laid during a 3-day period, were examined for the presence of salmonella. 7 of these eggs were sterile after the incubation period of 48 hours. Please remember that even though it was sterilized after an incubation period, 99% of microbes cannot be grown on a petri dish. That's why there's that common misconception of urine is sterile. Urine is not sterile. It's just we can't grow a lot of the microbes on a petri dish. If you run it through a PCR, you'll pick up lots and lots and lots of microbes that because it's just not favorable. So that's an interesting tidbit. Todd David says, so the oil from shedding is the most common vector, or is identified as probably the most likely? It's identified as probably the most likely from what I understand. I just remember when I first got my snakes, one of the first things that they told me straight off the bat was, after they shed, you need to clean the tank really, really well. Like, you should do that anyway, once a month, but you should clean the tank really well, make it all, clean it up, get rid of all of the bedding, make sure you sterilize it, because there is that chance of getting a salmonella poisoning. So, yeah, always wash your hands quite well after handling a fresh shed. Yeah, it's quite... And when you handle a fresh shed, you can feel it's really oily and really moist, and it feels weird. So, yeah, it wouldn't surprise me if there's a lot of bacteria running around that could get you really sick. And it wouldn't just be salmonella, it would also be E. coli, it would be any enterobacters. That could be enterobacter, meaning bacteria that can give you stomach problems. Yeah, so I wouldn't be overly keen on, let's say, licking a fresh shed or even an old shed. I always make sure that I give my hands a really good wash after I handle a new shed. Sam Chaos says, if you're careful, you can make the risk tank. I'm not sure what you mean by that. Probably a typo. Okay, so actually, going back to what Todd said about oil, going back to what I was saying earlier, besides Salmonella, other members of the family Enterobacteriaceae, such as Providentia retgeri, Escherichia, I'm sure I pronounced that wrong, Escherichia coli and Citrobacter frundi were isolated from 3 eggs. So they're all bacteria that make you sick. From investigations of chickens, it's known that Salmonella enteritidis, Latin's difficult, silently infects the ovaries of healthy appearing hens and contaminates the eggs before shells are formed, which is why they always say you should always cook eggs, never eat raw eggs, because hens can be healthy and look great and running around, but they've got a silent infection of their ovaries, and that's how it contaminates the eggs before the shells are formed. This leads to contaminated eggs in frequencies of about 1 to 7%, depending on the infection of the Salmonella strain. In our investigation, 65% of newborn snakes and 33% of non-fertilized eggs were colonized by Salmonella. This is a strong indication that the ovaries of the snakes are colonized also. Additionally, there is a striking difference between the eggs of hens and those of Bitis nasicornis and Bitis gabonica rhinoceros. Yeah, there is a little bit of a difference between those two particular eggs. Chickens have, they go on to explain it. These snakes do not form a shell around their eggs. So the newborns, as well as the yolk sac in the case of non-fertilized eggs, are only surrounded by a thin transparent membrane. So if you've ever seen a snake egg, they're actually kind of like squishy. They're not hard like a chicken egg. So they point out very rightfully, different Salmonella strains have been demonstrated capable of penetrating the vitelline membrane in hen eggs, which I think is that, when you crack open an egg, you've got that kind of weird white thing between the egg and the yolk, the shell and the yolk. That bit in there is the vitelline membrane. Therefore, it is conceivable they might be able to penetrate the membrane surrounding the snake's eggs. The relatively high percentage of primarily colonized newborns and non-fertilized eggs might be due to the fact that during the entire pregnancy, which is 9 months in the investigated snakes, Salmonella can potentially penetrate and colonize the eggs. This would also be a reasonable explanation for the variety of isolated Salmonella serotypes from the newborn. So basically says Latin is a dead language for a reason. Oh, okay. .nerd said, I think a spare tank would be a good idea for cleaning. Yeah, you can. When I clean, I usually have knot jar over. So I'm like, here you go, knot jar. Hold on to the snake while I clean out the tank. So he's sitting there playing with either Lilith or Thyla as here I am just scraping everything, cleaning my tank, cleaning this. This stuff gets freaking everywhere. Like, I cannot begin to tell you how much. It's ridiculous. I did a video on my other channel on snake handlers, from the Appalachian snake handlers, and throughout half of the video I could see this piece of stuff just attached to my hair, and I'm just like, well, I'm not going to edit that out. That's no point in doing that. But yeah, anyway, sorry. That's on a bit of a tangent. I love my snakes, but their bedding just gets freaking everywhere. But that's what I do. That's what I do. I'll get Thyla out. I'll get Thyla out after this because she's adorable, and I think you guys deserved that after hearing me try and butcher Latin. Okay, so the origin of Salmonella species in reptiles is not quite clear. In analogy to cattle and other warm-blooded stock, it could be assumed that Salmonella are introduced by food. However, the snakes included in this investigation have always been fed with laboratory mice and rats, which were guaranteed to be free of Salmonella species. Of course, it can be argued that colonization of pet reptiles occurs during the trade, where hundreds of reptiles are shipped together in small cages. Therefore, in the present study, newborn snakes were investigated. Am I even on... I am reading the wrong side. Okay, sorry, I'll start again. Okay. So it's been shown that the most prevalent Salmonella strains fecal samples from snakes may change over time. The mechanisms leading to these variations are hitherto unclear. Maybe some form of evolution could be like that, but basically they're not entirely sure how it happens. However, 2 of the newborn or female ones carried a serovar, which was found to be the prevailing one in their mother about 2 years ago. Although undetectable in this investigation, it must be assumed that the serovar, along with the others detected in her offspring, still is present in female number 1 in very small numbers. The frequency of these undetected serovars can be calculated to be at least being less than 2% of the entire Salmonella population in the snake's intestines. So it could be possible that as she changes, maybe her hormones or whatever, I don't know, this is me hypothesizing, I could be totally wrong, so don't take my word on this, but maybe as she grows or as she just life changes for her. Maybe 2 years ago they had a completely different setup for her. Her gut flora could change completely. Oh, Todd Davis says, fun fact, the membrane in chicken eggs can vary drastically in thickness depending on the calcium and vegetable proteins in the feed. Oh, interesting. Besides the frequency of certain Salmonella serovars, their ability to penetrate the membrane covering the newborn might also influence the transmission to the newborn. It's been shown that different serovars of S. entericus subspecies derived from rattlesnakes vary significantly in their ability to invade distinct cell lines. These people are bloody suicidal. I'm really impressed with their bravery. I mean, I wouldn't go anywhere near any of these snakes, to be honest with you. Fecal samples of the 2 maternal rhinoceros horned vipers were examined for the presence of Salmonella species immediately after they'd given birth to their offspring. From one snake, a total of 48 Salmonella colonies could be isolated and analysed. From the other female, only 5 colonies were detected and further analysed. All isolated Salmonella species belong to serogroup 3b. The serotyping results are summarised in Table 1, which I showed earlier. The Salmonella population of snake number 1 consisted of 3 different serotypes, of which a serotype was prevailing by far. However, none of the 9 colonized newborn of this female carried this particular serotype. So what they're saying is Salmonella this particular serotype was the most common for her, but none of the 9 colonized newborns of this female carried the serotype. The 2 other isolated serotypes were only found 4% and 2% respectively. 2 newborn of female 1 were colonized with serovar, which previously found in the snake. And again, they often repeat themselves, I've noticed, when you're reading the results. So the population analysis of snake number 2 showed a different pattern. The Salmonella constituted only a small proportion of the entire enteric fauna of the snake. However, the few Salmonella which were detectable belonged to 2 different serovars, which contributed nearly equally to the population. Oh, hang on, I should have... Give me a moment. Sorry, I'll start again. The population analysis of snake number 2 showed a different pattern. Salmonella constituted of only a small proportion of the entire enteric fauna of the snake. However, few salmonella were detectable belonged to 2 different serovars, of which contributed nearly equally to the population. Of the 5 colonies which were examined, 3 belonged to the serotype 3B4Z10Z and 2 to serotype 3B48IZ. Both serotypes were also detected in the feces of 2 and 1 newborn, respectively. Yeah. So I'm hearing .nerd, maybe they wore masks when taking those samples. Unfortunately, I don't think that, well, they might have. But this was, when was this published? This was published in 2006, so it's a while ago. They probably wore gloves for the rhino viper's venom is very nasty to the body. Yeah, most venom is. Yeah, the most venomous snake on the planet is an Australian one, of course, because it's Australia, and it is the inland taipan, and it is so toxic to humans that even simply handling it can send people into a coma. Like, just from the absorptivity into the body can cause them to go into coma. That blows my mind. J.R. Eldridge, I'm late, you're going to have to repeat everything. Salmonella is found in snakes. There we go. Yeah, so what I said before, I was reading a different column of this paper. I'm sorry, I ran out of milk. I haven't had coffee. So the origin of the Salmonella species in reptiles is not quite clear. In an analogy to cattle and other warm-blooded stock, it could be assumed that the Salmonella are introduced by food. However, the snakes included in this investigation have always been fed with laboratory mice and rats, which were guaranteed to be free of Salmonella species. Of course, it can be argued that colonization of pet reptiles occurs during the trade where hundreds of reptiles are shipped together in small cages. Therefore, in the present study, newborn snakes were investigated which were born in captivity and which were singularly kept under excellent hygienic conditions. The present investigation demonstrates that a significant number of snakes become colonized with Salmonella during pregnancy. However, this seems not to be the only mechanism, because a study on adult B. nasicornis has revealed that literally all examined individuals were positive for Salmonella. As ground-dwelling snakes, B. nasicornis, as well as B. gabonica rhinoceros, have a certain risk of coming into contact with Salmonella contaminated feces of other animals. This can be considered another important method of transmission because arboreal snakes, whose risk of close encounter with Salmonella contamination is lower, have been found to be less frequently colonized. Yes. So that was it. I thought that was pretty interesting. They found that that the transmission of Salmonella into a reptile's offspring comes from the mother, both with viviparous and oviparous species of snake. In regards to my girls, I don't know who their mothers are, so I can't tell you that. But from what I'm reading, the chances are that they're highly likely to be positive for Salmonella. So luckily, I haven't got sick yet. I do clean the tank quite well every month, or as soon as I do the spot clean because they're quite clean animals. So, with this particular substrate, it has a dye in it, a AV dye or something like that. And basically what happens with this dye is it changes color when it comes in contact with ammonia. Is it ammonia? I think it's ammonia. Basically what happens is it changes color when it comes in contact with urea. And so I did some experiments and I found that if you put it with a base, it will change color, and that's how you can find out if they peed. Is you look in it. If it's bright yellow, it is that's being contaminated with urine, and then you just take it out of the tank. J.R. Eldritch says, so the Bible is right. Females are really icky. Dot Nerd says, I've watched this one guy who had a show being stung by insects. Last one he did was a centipede. Can't imagine him doing snakes though. Why would you do that? Why? No worry, Bill. Thanks for coming. I really appreciate it. This one's a shorter one. It doesn't require me going, I don't quite understand it. This is a fairly easy one to understand. And yeah, I just thought it was really cool. Anyway, I'm going to go and get Thyla out. So give me a moment. I'll get her out. Hello. Can I get you out? Oh, you're so cute. I know. You know what to do? That's good, isn't it? Here she is. So there we go. Here's the little girl. She's adorable, isn't she? She's like, what are you doing? I should be asleep. Yes, you should be asleep. I need to take you out and say hello to the sun. I don't know if you can see particularly well, but see how she kind of here and then she goes in quite quickly? To me, that means that she's probably getting close to doing a poo. So I get to go and sterilize a jar. I need to go and grab that fairly shortly. To me because it's not so pronounced when she doesn't need to do a poo. Anyway, good fun. Oh, MJB says we used to keep blue tongue lizards, not quite the same as snakes, but we ended up nursing one for years after she got an infected bite wound on her mouth from the male. Oh, I didn't end up getting it because I didn't have $1,200 or $1,200. So closer to $2,000 after you include all the stuff that you need to buy. A hypermelanistic blue tongue lizard. Well, hypermelanistic meaning that it's all black. It was cool. And they were selling it for $1,200 and I was like, I really want it. But I was surprised. They said last time they had one of those in, it took about 2 years to sell them because it was, it was just too expensive. And then when I went in the next time, they were like, oh no, we sold it. And I was like, well, that was a good thing. That was a good thing because I was really, really close to getting it. Even though I didn't have the money, I didn't have the space, but it was so cool. Hi, Alias, how you going? We just finished up. This one was a quick one. So what's happening? Where am I up to in regards to my project is I'm going to be seeing my person on Tuesday, and we're going to be going through applying for ethics, because even though the snakes themselves have nothing will change in their life at all in regards to being handled, being fed, that sort of stuff, you still need ethics approval for it. So we're going to be doing applying for ethics. Hopefully I'll be able to start plating up and figuring out when I can plate up the things that we need to do. So that's where I'm at. I've got to go through and do all my safety because you got to do that every year. And yeah, so that's where I'm up to with my research project. And yeah, hopefully all that will be going on. But that's basically it. Oh, hello. Okay, does anyone have any questions? Does anybody have any questions or comments? Have the snakes signed consent forms? No, they haven't. So they don't know that I am telling everyone about their bowel motions. I honestly don't think, as I said, the ethics committee would be terribly upset about. Please don't go on the keyboard, sweetie. Yeah, I don't think anyone, the ethics department will be upset about this in any way, because, as I said, nothing will change for my snakes. They'll still be loved and cared for and have good, be pretty much stress-free life. Unless, of course, they're like, I don't want to go out of the tank. That's probably the biggest stress that they go through. They don't have to worry about hunting or anything like that. So if I start typing random stuff, it's Thyla. She's climbing all over my keyboard. Okay, well, I do think that's everything. Thank you everyone for joining. I really appreciate that. This is again J.R. Eldridge. I'm envious. I can't get anyone to listen to me about my bowel movements. I can't either. Yeah, not many people want to know about my bowel movements. Yes, but thank you everyone for coming. I really appreciate that you guys are helping me keep on top of things. And I really appreciate all of your support for it. And get to learn with me, get to watch how I go. And I hope everyone stays safe and healthy. Go get your vaccines. If you can, go get your vaccines. And I'll talk to you later. And don't forget, scandium, iodine, nitrogen, and cerium.